This video will show you how to operate in the basic interface and perform searches in the TRANSFAC database. To open the TRANSFAC database, please log in at portal.genexplain.com and select TRANSFAC. In the interface of the TRANSFAC database, you will find three tabs at the start page. The My Data tab will show you all the data and analysis result which you have saved to your account. The Tools tab will show you gene regulation analysis and other tools available in TRANSFAC. Tools which are not active, like network analysis, require additional subscription to other databases such as Proteome or TransPath. The Search tab allows you to perform database searches, so let's start a new search. As an example, let's search for the STAT transcription factor. As you can see, 207 results were found for the STAT among all genes and proteins in the TRANSFAC database. However, this also includes some less specific results where STAT was found in keywords rather than in the gene or protein name. To narrow our search, we can click on the View More search options. Let's search for transcription factors, and only in mammalian species. If we execute search now, there will be only 52 total results for the STAT factor. As you can see, we have results for human, mouse, and rat, and some other mammals. We can sort this table by species. We can also save these results if we need it. We can now search for the transcription factors of these genes by clicking the Binding Factors for Gene Search. The result will open in a new tab, and it will show us the transcription factors which have been experimentally proven to bind to these genes in our list. We have a total of 50 binding factors found. These are factors for which at least one binding site has been shown in any of the STAT genes from the input list, including orthologs, splice variants, and complexes. And now, let's go back to our search results and select the Gene Bound by Factor option. The result of this search will show you the genes which have binding sites for any of the STAT factors. When we click on a name of a gene, we get a locus report open in a new tab. As you can see, this report provides a schematic view of the binding sites and the transcription factors that were shown to bind to this gene. Now let's go back to the start page. The main search supports the most frequently used search options, but there are additional search tools included which allow to address also more specific questions. For this, let's select Build a Custom Search option. We can now perform the same search which we have just done in 2 steps simply by 1 step. For this, let's search for a gene which has a factor binding site named STAT and press on the search button. As you can see, we have a total of 466 genes and proteins found. Now, let's narrow our search by adding a new transcription factor. So, we're searching for a gene which simultaneously has a factor binding site named STAT and a factor binding site named CRAB. If we execute search now, we have a total of 57 genes and proteins found. These genes simultaneously have binding sites for STAT and CRAB transcription factors. We can save any of these genes by simply selecting them and pressing the Save these results button. Now our results have been successfully saved. Another type of search in the TRANSFAC database is the ontology search. The ontology search tool allows you to search for genes, diseases, drugs, microRNAs, and variants with common characteristics. As an example, we are going to search for genes expressed in active T cells but not in resting T cells. For this, we first are going to select Expression and search for T cells. We have 10 categories found for T cells. Let's select active T cells. The two letters EX abbreviation refers to expression. If you are interested in other abbreviations, they can be found in the Help and the User Manual link. So, we are selecting genes which have expression of active T cells. As you can see, we have a total of 218 genes found. We can view all of these genes by pressing this button. We can now exclude from our search those genes which are associated with resting T cells expressions. For that, let's select resting T cells expression and put is not operated and execute search. As you can see, we have 111 genes found. We can view the full list by pressing this button. We can export these results for further investigations or save them in the account data. This video showed you how to operate in the basic interface and perform search in the TRANSFAC database. Watch our further videos for other TRANSFAC options. If you have any questions, please contact us by email info@genexplain.com. We hope you find this video useful and thank you for watching.